続きまして、弊社アメリカ本社からのですね、クリニカル&トランスレーショナルマーケティングマネージャーのラウラ・ウリベより弊社のテクノロジーについてご紹介させていただきます。
Okay. So, good afternoon, everyone. Thank you for joining us today. I would like to talk to you a little bit about how Affymetrix is investing to bring you and the patients some very interesting genomic tools to improve the way we diagnose disease. So I will try to focus my talk today on two of the main applications we have been working and investing in developing products for improving the diagnosis, specifically in cytogenetics and carrier screening, and in cancer genetics. So this slide here shows you a paper from the Deciphering Developmental Disorders Study. And one of the first statements of this paper shows the actual frustrations some pediatricians and clinicians are facing today. That means they don't have all the times an answer to give to their patients. So that's a significant burden, not only for the clinician, but also for the families and the health services, as it is hard for them not to get an answer on what's going on with the patient. Cytogeneticists have a deep interest in providing the highest level of patient care. So the problem is with current technologies, there is a poor diagnostic yield. 65 to 80% of intellectual disability cases are of unknown cause. And sometimes we don't know what's the cause because we don't have the tools, the proper tools to analyze what's going on. Patients with unexplained intellectual disabilities are usually screened with karyotyping and FISH, and the combined diagnostic yield of these two techniques is approximately 5 to 10%. That means that a clinical diagnosis is lacking in the majority of these patients. That complicates the treatment and, of course, the genetic counseling of the family. So since 2010, after using microarray technologies for a while, mainly for research, the American College of Medical Genetics stated that we should recommend karyotyping for microarray testing. After this recommendation was made public, there have been a lot of studies showing the importance and the application of microarrays in other studies, such as prenatal testing, autism analysis, and recently there was another study that is today being completed. It's done again with a higher number of samples, where they are using chromosomal microarray analysis for prenatal testing. They are showing microarrays give a better understanding of the probable pathologies a product may have, and they can give a better answer to the patients compared to the techniques that are used today. That it's mainly done by FISH and karyotyping, of course. So what Affymetrix has been working since a couple of years ago is with the Cytoscan platform. We call it a Cytoscan platform because we have actually two arrays. We have a high-density array and a low-density array, so it's not just one product. It's a complete platform. And we also call it a platform as we provide you with the microarray, the reagents, the system, and the software to do data analysis. The platform has some very interesting specifications. It has a gene-level coverage, meaning we can analyze more than 20,000 RevSeq genes. It has 2.6 million empirically selected probes. These probes are composed of copy number probes and SNP probes. It has a copy number resolution of about 25 to 50 kbs across the genome. And as we are adding some SNP probes, we cover other things such as regions. We can detect regions of absence of heterozygosity. We can detect mosaicism and other things that can only be possible to analyze when you are using SNP and not only copy number probes. It's a product that has been screened and used with multiple sample types, such as blood, saliva, bone marrow, products of conception. So it's a product that many labs around the world have been using to do a postnatal analysis. Why do we think our product is superior and provides a better understanding of what's going on compared to other technologies such as karyotyping? Well, this is a very brief example here. What you see here is a karyotype of a sample. You can see the sample has multiple aberrations, a trisomy in chromosome 8, another one here in chromosome 22. And where we have the arrow here, we see there's a gain in chromosome 7. So if you look at the karyotype, it's really difficult to understand where this extra material from chromosome 7 is coming from. But this sample was run and analyzed with our cytoscan array. And this is the typical view of a result of the array. And we have several tracks that give us different information. So this one here is the log-to ratio, and this one is the copy number, the smooth signal that give us an idea of the copy number of this particular chromosome. So if you look here in the smooth signal, we can see there's a shift in chromosome number 1. So we are seeing there's a gain, there's a duplication of chromosome 1, but we don't see that here. So probably this material that's coming, that we are seeing here in chromosome 7, it's coming from this region of chromosome number 1. So that's one of the examples on how microarrays can be very useful and help us understand more of what's going on a sample. We have a very, very dense coverage in a platform. This is just an example of the differences of our product with other products that are already in the market. So this is the Agilent 180K ISCA array. It's an array that was customly designed with the ISCA genes. And you can see here we have a very, very dense coverage in all the array compared to the other platforms. The other platforms have a lot of spaces in between, as for example Illumina only uses SNPs, and Agilent has more density in particular genes that are important because it's a focused design. What's the importance of having such a high density? Of why do we think having a high density array is helpful? Well, this is just an example of how with cytoscan array we were able to identify this 41 kb deletion in this particular sample, and this deletion is right where this particular exon of MBD5 gene is located. So what's the importance? Recently, this gene has been linked to autism. So other platforms with very low density will definitely have missed this particular imbalance here. And our high density platform was very, very, very robust to detect this particular change, which is definitely now associated to disease. Another powerful thing about Affymetrix microarrays, and specifically of our cytoscan platform, is the use of SNPs. SNPs help out to understand several things, such as allelic-specific changes, maternal contamination, low-level mosaicism, triploidy. They are useful to improve the breakpoint determination. Trio consistency. As you have genotyping, you can analyze the parents and be sure you are getting, you can analyze, there's a trio consistency, that the parents you are analyzing are the parents of the child with the problem. And it also is useful to confirm copy number events. This is a recent paper that was published, I think it was published in April this year, They compared 3 different platforms, one low-density array, a 44K array from Agilent, a mid-density array, the 180K array, and they also used our Cytoscan HD. So the title of the paper was Density Matters when they are trying to understand copy number variation and copy neutral aberrations. So this figure here shows a deletion that was detected by the 3 platforms. So if you look at the low density array here, it is detecting quite well the 648 kb aberration here. The mid-density array, you can also see there's a detection here, but it's not so clear, probably because this array has more probes in other regions of the genome. But here it's an example of the power of having a high-density platform that combines copy number probes and SNP probes. You can see here the copy number probes detect clearly the deletion, and you have here the allele tracks where you can also detect there's 3 lines when it's a copy number normal or copy number 2, and you can see 2 lines here when there's a deleted region. So we have a confirmation of this by using these 2 types of probes. Plus, this paper also detected that the low density platform overestimated the regions of absence of heterozygosity, meaning as they don't have enough probes, enough SNP probes all along the region, it was very difficult for these 2 platforms to detect accurately the regions of LOH in the samples. So these examples I have talked about are mainly on constitutional or postnatal samples, but today arrays are also being used for the analysis and the diagnosis of leukemia. One of the main problems of analyzing leukemia sample is that culture often fails. Cell culture of leukemia samples is very difficult to do. So they don't get an answer because they cannot culture the cells. With a microarray, you don't need to do this cell culture, you just need the DNA. You get exceptional resolution. You can identify very small CMBs and accurately detect breakpoints. It also detects copy number change, copy neutral LOH, and this is a very important thing in leukemias. 50 to 60% of leukemias have copy number neutral loss of heterozygosity events. You can also detect low-level mosaicism, and it's a cost-effective alternative fish. Why do we say it's a cost-effective alternative to fish? Because you don't need to do 1 fish probe, and if it fails, do another one. You just do 1 experiment, analyze the whole genome, and that's mainly it. So this is an example of how Cytoscan has been used for a leukemia case, a myelodysplastic syndrome case. These are 2 cases where the patients were diagnosed with a particular kind of myelodysplastic disorder, and their cytogenetic finding was completely normal. They did not find anything by doing karyotyping. But when they run the samples in the array, what they saw was a quite huge deletion, a 2.6 megabases deletion in one of the samples. This is I'm just showing one of the samples. And you can see here's how the software tells us there's a deleted region. We see the deletion right here. And all the pink tracks here that are shown by the software are genes. So this deletion is located where the RUNX1 gene is. So this gene has been related to poor diagnosis in leukemia patients. So they are resistant to therapy and they have an inferior outcome. So this is just an example of how microarrays help to understand and to actually give a prognosis to a patient where the traditional techniques show the patient was actually normal. So as we are moving toward cancer, I just want to stress out the importance and why we are also investing a lot of time and energy on providing products to study cancer. It's the 2nd leading cause of death worldwide. 8 million people die of cancer every day. There are 2 million cancer diagnoses expected this year just in the US. 900 people die of cancer every hour. 1/2 of men will get cancer in their lifetime and 1/3 of women. So we know cancer is important, we know cancer is a health issue, but why haven't we been able to get or understand cancer better? It's a very complex disease. It has a depth of complexity, so we need to consider multiple markers when studying cancer, and we have to consider different genomic parameters, meaning cancer has been related to mutations, but cancer has also been related to copy number changes. You can see here, this is an example of a copy number of a karyotype of a cancer sample. There are too many copy number changes. There are a lot of duplications in entire chromosomes. So we need to provide and do massive parallel testing to get a better understanding and a more comprehensive understanding of the cancer genome. And as you know, when patients are diagnosed, they usually get biopsied, and all the biopsies go to are paraffin embedded. So 80 to 90% of all solid tumor samples go through this process. These are irreplaceable clinical materials because we have been collected this type of samples for many years. We have extensive clinical history of these samples, as we know the pathology, we know the diagnosis. For some of them, we know the progression, the treatment, and the survival of the patient. There are more than 1 billion tissue samples archived in hospitals in paraffin blocks. So why haven't we been able to analyze the genome of these samples? Well, FFPE samples, when you do the process of embedding the sample in paraffin, DNA gets degraded. It suffers from a variable fixing, and there are variable collection methods, and depending on the method, it's the extent of degradation of DNA. And they have a lot of problems, and the outcome of all these issues is we get very poor quality results when we extract DNA. So we have recently launched this OncoScan product. It's an OncoScan, it's called OncoScan FFP assay. So we just need 80 nanograms of DNA extracted from paraffin. We have validated this product with samples that are 10 years old. It works very well with highly degraded DNA. You process the sample, and after 48 hours, you get the results. It works with the existing Affymetrix platform. It has a very good resolution for copy number changes on 900, specifically 900 cancer genes, and it has a very good resolution on the whole genome. We also have SNPs in this particular array, so we are also able to detect LOH events. And we added some probes to detect 20 of the most important clinically actionable somatic mutations. This is an example of how the data looks like on the OncoScan array. Each of the colors here you see is one chromosome. All the dots are the probes of the array. So this is an example of a breast cancer patient. Breast cancer patients are usually screened for the HER2 gene to see how many copies of the HER2 gene are in the patient sample, because if the patient has many copies of the HER2 gene, they will get a particular treatment. They will get prescribed with Herceptin. So having a patient with this amplification will give a prognosis and will actually provide the doctor an idea to how to treat the patient. So the amplification is on chromosome 17. HER2 is on chromosome 17. So you can see here, we can definitely detect this region with the array, and there's also this deletion on chromosome 8. So what they usually do with these samples is they run a FISH probe, and if we think about it, if this sample was done with FISH, it would have come positive for HER2. So the patient would be prescribed with Herceptin. But this is a genome of another patient that if we only did FISH, we would definitely have caught, you can see here, chromosome 17. It also has an amplification on chromosome 17. But look at the rest of the genome of this patient. It has too many aberrations. There are gains and losses all around the genome. And this is probably a patient that will have a very reserved prognosis. So do you think if we only run the FISH probe, this patient would be, classified the same as the patient we saw before? But the genome of this patient is definitely very different. So that's why we think doing a whole copy number analysis is really important to understand what's happening with cancer samples. This is just another example. This group did some analysis to understand how could they classify gastrointestinal tumor samples. And they thought, they started doing analysis of these two mutations, the KIT and PDGFRA gene. And what they saw is they decided not only to do a mutation analysis, but they also did a copy number analysis of all the samples they had. And they were able to understand and to look at the data, the clinical data, and they were able to see that copy number changes contributed significantly to the development and progression of gastrointestinal stromal tumors. So they saw, analyzing not only the mutations, but also the copy number changes, they were able to classify patients much better and to understand how the prognosis of the patient was going to be. This is just another example. This is from a group in Australia. This group studied ovarian cancer, which is the 7th cause of death in women worldwide. The problem of ovarian cancer is that when women arrive to, when they already have the cancer, the tumor is usually very developed, is usually detected in a very late stage. Because there's lack of knowledge of the molecular events that initiate disease. So what this group wanted to do is to analyze benign samples, benign tumor samples from patients with that had these ovarian cysts, and they wanted to understand if there was any event that could probably be a biomarker of initiation of malignancy. So what they did is they sliced the FFPE samples, and they found, and they tried to classify endothelial tissue and fibroblasts in the samples, and what they found were very specific copy number changes in the fibroblast cells that probably were the ones that were initiating the molecular response to particular oncogenes, and these genes were delivering signals of growth that will probably also affecting the epithelial cells, and then converting these cells into malignancy. And then it was like, it is a very interesting reciprocal model that probably is what could explain the initiation of ovarian cancer. And this was only possible to do because they were able to analyze very small pieces of samples and of tumors that were embedded in paraffin. Another very interesting phenomena that has been Another interesting thing that can be detected by this genomic array is chromothripsis. Chromothripsis is a chaotic rearrangement that happens in chromosomes in cancer samples. So there's a shattering in the chromosome, that the chromosome breaks into pieces, rearranges, and comes together again. And this possibly is something that is it's really common in some types of cancer, and it's related, obviously, to a poor prognosis. This is how OncoScan can detect chemothripsis. You can see here, there are some regions in this particular chromosome that look like very, very defined lines. So this is what's going on. This chromosome has some small deletions there or some losses there, and this is probably how chromathripsis can be detected in a cancer sample using the microarray. So in summary, we have this new product that provides you not only with a very powerful, well-designed array, we also have reagents that can process FFPE samples. We will provide a software for analysis that is free of charge, and it's and the instrumentation that has been around in your laboratories can be used for this particular array. Last but not least, I will talk briefly about some of the new gene expression developments we have. As you know, gene expression has been used for a while for disease classification in cancer, biomarker discovery, and treatment selection. But today, the trends in gene expression studies are that we need to understand better the transcriptome and understand better the biology. We need to look at everything that's relevant. We need to understand alternative splicing. We need to understand how the expression is controlled. So non-coding RNAs, including microarrays, need to be understood. And we also need to detect rare transcripts. We know the human genome has around 20,000 genes, but the proteome is highly complex, and we have many more proteins compared to the number of genes existing. And this is probably one of the reasons we have such a diversity on proteins, is because we have the alternative splicing events. So we recently developed the Human Transcriptome Array. It's a new exon and splice junction microarray that includes both non-coding and coding gene models. It has the most comprehensive view of the transcriptome, and we also have recently designed a free-of-charge software that can be used with this particular array. So this is an example of the coverage of the HTA array. This is a gene, and you can see many databases are telling us there are different transcript forms for this particular gene. So we are covering every single one of the described transcripts, and we are also covering the junction regions with 4 probes, each junction region in the array. I will not go through this one. I will just want to show you this particular slide that shows a little bit how the array is designed. This is the gene. You can see the exons here. We have around 20 probes per exon, and we have around 4 probes per splice junction region. So this is how we are really covering very, very comprehensively all genes in the transcriptome. This is just the numbers of how many probes we have per transcript clusters, per exon cluster, and per splice junction regions. So the importance of this array is you can not only detect gene level changes, you can detect exon level or isoform changes, and you can also detect link RNAs, which are now a very interesting new set of RNAs that are related to the to how transcription is controlled. That is all that I had to talk to you. Thank you, and I am open to questions. So before I leave, I want to introduce our next speaker. Our next speaker is Dr. Adam Smith. Adam was a clinical cytogeneticist in the Hospital for Sick Children in Toronto. Then he was recruited by the British Columbia Cancer Agency as a clinical cytogeneticist in the Cancer Genetic Laboratories. He recently, a few years ago, moved to Brazil, where he is a scientist at the Pelé Research Institute, part of the Hospital Pequeno Príncipe. And he's also a clinical assistant professor at the University of British Columbia, and also he's doing some he's doing some advising work for a very state-of-the-art diagnostic lab in Brazil called Mantis Advanced Diagnostics in Curitiba, Brazil. So I would like to thank Adam for being here with us and leave you the floor.